Discuss about the molecular basis of inheritance. We have discussed about principles of inheritance and variations in the previous class. Where factors we spoke about genes, but we did not speak about how the gene is made up of. What is the gene? What is it composed of? All these factors we are going to discuss in this molecular basis of inheritance. Already you have studied in first year biomolecules. You might have studied about DNA and RNA. What are the differences between DNA and RNA? First point is what type of sugar DNA has. Deoxyribose sugar DNA has and RNA has ribose sugar. DNA has two functions. Can you tell me what are those two functions? Autocatalytic function and heterocatalytic function. So autocatalytic, this part of it, DNA synthesizing itself, we call it as autocatalytic function. DNA synthesizing DNA, we call it as autocatalytic function. Heterocatalytic function of DNA is DNA synthesizing RNA. DNA synthesizing RNA, we call it as heterocatalytic function. So they have two functions. Okay. Majority of organisms, DNA is the genetic material. A few organisms like viruses, RNA is the genetic material. Majority of the organisms, DNA is the genetic material. DNA is of only one type. RNA is of three types. What are the three types of RNA? Messenger RNA, ribosomal RNA, tRNA or transfer RNA. So these are the three types of RNA. DNA has thymine, RNA has uracil. DNA has thymine, RNA has uracil. DNA is a double helical structure. RNA is a single stranded, except at loops they might be double stranded as in case of tRNA. So these are some major differences between DNA and RNA. Deoxyribose nucleic acid, ribose nucleic acid, whenever we are talking about it. Okay, so we have already discussed about the types of nucleic acid, ribose nucleic acid, deoxyribose nucleic acid. RNA has additional role apart from being a genetic material in a few viruses. They also have additional role like adapter, tRNA, structural molecule. It can also be catalytic molecule. One of the important aspects of molecular biology is central dogma of molecular biology. Dogma andre unchanged principle. Unchanged principle. So DNA synthesizing itself, we call it as autocatalytic function, replication. Okay, that's the reason I put a circle. DNA undergoes replication to produce a copy of DNA. This DNA undergoes transcription to produce messenger RNA, and messenger RNA undergoes translation. They have a message, coded message. This coded message is translated into proteins. They are translated into amino acids. Polymer of amino acids they form the protein. So the central dogma of molecular biology involves replication, transcription, translation. One more difference between DNA and RNA, you can say it as DNA synthesizes mRNA, we call it as transcription. RNA synthesizes DNA, we call it as reverse transcription. If RNA has a genetic material, it synthesizes DNA, we call that process as reverse transcription. That is the differences between DNA and RNA. Okay. Apart from that, what are the nitrogenous bases found in DNA? Adenine, guanine, cytosine, thymine. What are the nitrogenous bases found in RNA? Adenine, guanine, cytosine, uracil. Instead of thymine, they have uracil. That's how you know that it is an RNA. Okay. Now today it is an era of genomics. What is genome? What is genome? Sum total of genes present in haploid set of chromosomes. a human genome project ಅಂತ ಹೇಳ್ತೀವಿ. They have already got the entire information of human genome. Human beings, they have 46 chromosomes. 23 chromosome ಅಲ್ಲಿ they have collected the data. The sum total of genes present in 23 chromosomes is human genome. Okay, haploid set of chromosomes. Innೊಂದು set ಯಾಕೆ ತಗೊಳ್ಳಿಲ್ಲ? It is the same repetition. Sum total of genes present in haploid set of chromosomes, we call it as genome. Today it's an era of genomics. Tomorrow you have some sickness or some abnormality. The root cause, the DNA can be rectified by gene therapy. ಈಗ ಕೀಮೋಥೆರಪಿ ಇದೆ, ನಿಮ್ಗೇನೋ ಕಾಯಿಲೆ ಬಂತು ಅಂದ್ರೆ ಏನು ಮಾಡ್ತೀರಾ? ಔಷಧಿ ತಗೊಳ್ತೀರಾ in the form of tablets, syrup or capsules, isn't it? So later stages, another 10 or 15 years, it might be gene therapy. Already gene therapy is taking place. Root cause itself is rectified. So many progress is taking place. Today it's an era of genomics. The determination of complete nucleotide sequence of human genome has happened. Okay, so that database we have now. So the DNA, let us understand what the DNA is. DNA is defined as a long polymer of deoxyribonucleotides. What are the components of DNA? Deoxyribose nucleic acid, what is it made up of? They are made up of polymer of nucleotides. Then what is nucleotide? Nucleotide is composed of nitrogenous base, sugar, pentose sugar and phosphate group. All these three together we call it as nucleotide. If we are going to consider only nitrogenous base and pentose sugar, what can we call this nucleoside? We call it as nucleoside. See, nitrogenous base plus pentose sugar, we call it as nucleoside. If it has phosphate group, we call it as nucleotide. So replacing equation 1 in equation 2, nucleotide can also be written as nucleotide equals nucleoside plus phosphate. Can't we do that? We can also write it as nucleoside plus phosphate. That is another definition of nucleotide. Understand about it. Whenever I am using the term nucleoside, what am I talking about? Nitrogenous base and sugar. If I use the term nucleotide, I am talking about nitrogenous base, sugar and phosphate group. These are the components of nucleotide. One more question here about it. Now the nitrogenous base is attached to the pentose sugar by N-glycosidic linkage. What is the linkage called as? N-glycosidic linkage. Now pentose sugar is attached to phosphate group by what linkage? Phosphoester linkage. Diester or phosphoester linkage. Okay. So this is what you have to remember regarding the linkages. That's the reason I gave it in the formula part. Nitrogenous bases are of two types. Single chain, double chain. Okay. So the ribose, the pentose sugar, you know, ribose and deoxyribose. Nitrogenous bases are of two types, purines and pyrimidines. Purines are the examples are adenine and guanine, and pyrimidines, you can remember it as CUT, cut, cytosine, uracil or thymine. In RNA it is uracil, in DNA it is thymine. So pyrimidines are cytosine and thymine in DNA. In RNA it is cytosine and uracil. So RNA you find this uracil, in DNA you find the thymine. In polynucleotide another ಮುಂಚೆ dinucleotide ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. One nucleotide getting attached to another nucleotide, we call it as dinucleotides. And that linkage is from 3' to 5' end, the linkage that is going to form as diphosphoester linkage, because two nucleotides are there and they are going to form dinucleotides. To this dinucleotides, if there are more nucleotides added, we get polynucleotides, which is also called as polymer of nucleotides. Polymer of nucleotides. So thus we can define DNA is a long polymer of deoxyribonucleotides. It's a long polymer of deoxyribonucleotides. That is what is the definition of DNA. Now polymer at one end, if you look into 3' and 5' end, polymer at one end has a free phosphate group, so that we call it as the 5' end of the ribose sugar. At the other end, 3' end and OH group irutte, and we refer to this as 3' end. So there are anti-parallel strands. When we come to the structure of DNA, I'll be telling you. One strand is in the direction from 5' to 3', another strand is in the direction of 3' to 5'. Anti-parallel strands are there. That we'll discuss later. See, this entire nucleoside, nitrogenous base and pentose sugar, they are the backbone of the structure of DNA. They are the backbone of the DNA structure. Okay, remember about that. DNA was, nucleic acids were discovered by Frederick Meister. Okay, DNA was studied by Frederick Meister in 1869. But there was no progress further. He called this nucleic acids as nuclein. He did not even term it as nucleic acid. Okay, instead he was using the term nuclein. After nearly 1953, recent subject it is, they discovered, they analyzed, they put forth the structure of DNA, double helical model of DNA, double helix model of DNA. This was put forth by Watson and Crick. But for Watson and Crick to put forth this double helical model of DNA, they required the information that was gathered by Wilkins and Franklin. Okay? Wilkins, Maurice Wilkins and Rosalind Franklin, they made an X-ray diffraction study of nucleic acid, DNA. Based on this information, James Watson and Frederick Crick, they put forth the structure of DNA, that is double helical model of DNA. But they required the work of Wilkins and Franklin. Watson and Crick required the work of Wilkins and Franklin to put forth the structure of DNA in 1953. They got Nobel Prize for this epoch work. So molecular biology is very recent in origin. 1953 we are studying molecular biology. After understanding the structure of DNA, lot of progress has happened. So there is also a scientist, Ravi.G, who put for base equivalence rule. According to Ravi.G base equivalence rule, for a given structure of DNA, any fragment or length of DNA, the ratio of adenine and thymine and the ratio of cytosine and guanine, they are going to be constant and equal to 1. See, adenine base pairs with thymine, the bond is double bond. Cytosine base pairs with guanine, the bond is triple bond. You should know about that and discuss about that when we come to the structure of DNA. Okay? So for any fragment of DNA or for any double-stranded DNA, the ratio of adenine and guanine and the ratio of cytosine and thymine are constant and equal to 1. Okay? Are constant and equal to 1. This we call it as Ravi.G base equivalence rule. We can also put it in simple word. For any length of DNA or fragment of DNA, the amount of adenine is equal to the amount of thymine and the amount of cytosine is equal to the amount of guanine. Okay? Because adenine and guanine are complementary in base pairing. They are complementary in base pairing. Sorry, adenine and thymine are complementary in base pairing. Cytosine and guanine are complementary in base pairing. The bond between adenine and thymine will be double bond. The bond between cytosine and guanine will be triple bond. So all those we'll be studying in detail later when we come to the structure of DNA, double helical structure of DNA. And also what they noticed was polynucleotide chains that are there, they are complementary to each other. If I know this complement, the sequence of this green color, let me just assume CAA, GCA. If I know this complementary strand base sequences, I can predict another strand base sequences. What is the complementary base pairing of C? It would be G. What is the complementary base pairing of A? It would be T. So you can predict the another strand. So that is how the parental strands in the LL.B. Semi-conservative mode of replication. When we come to that concept, I'll tell you. So the parental strands is separate out and the newly formed strand will have a complementary sequence to the parental strand. The parental strand base pairing and the newly formed DNA base pairing, they are identical copies. They are identical copies. The newly synthesized strand, parental strand ಏನಂತೀವಿ template strand ಅಂತೀವಿ. ಹೊಸದಾಗಿ synthesize ಆಗುವಂತ strand ನ daughter strands ಅಂತಾರೆ. Template strand ಇಲ್ಲಿ white chalk ಅಲ್ಲಿ ಬರ್ದಿದೆ ನೋಡಿ daughter strands. Okay, this is semi-conservative mode of replication of DNA, which we'll be discussing in the later classes. This is what you have to know for the introduction of DNA.